Welcome back to the final session for today by Dr. Anupama Krishnamurthy on the topic, why does the past hold the key to the future? I don't know how many of you have heard about the paleobotany or paleosciences or palynology. It's one of the most exciting branches of historical sciences based on evidences. And of course, there are very few experts working across the country in BSIP and one in French Institute. And it's my pleasure welcoming Dr. Anupama Krishnamurthy, one of the renowned Palynologist and Paleoecologist in the country. She is currently working in the Department of Ecology at the French Institute of Pondicherry. Since more than 25 years, she has been carrying out research in the fields of paleo-vegetation reconstruction, pollination ecology, and aerobiology, employing pollen grains as the key tool. Her expertise also includes non-pollen palynomorphs such as phytoliths that the team has applied in archaeological and other contexts in South India. Building a repertoire of phytolith slides from the extant plants in tropical South India and Sri Lanka along the same lines as Tanigamani pollen slide collection at the IFP is a current preoccupation in addition to a project on pollination and pollinators of South India. She works in multidisciplinary teams comprised of plant ecologists, archaeologists, and sociologists. She is a recognized supervisor of research and she is recognized supervisor of research and PhD level by Pondicherry University and also co-supervised and supervised doctoral and postdoctoral students on various aspects of quaternary palynology and melisopaleontology, including quantitative modeling based approaches in pollen-based land cover reconstruction. In addition to this, she has also supervised many masters, MPhils, and undergraduate students from India and abroad. She is currently the president Humans and Biosphere Commission of INQEA and a member of Page's Land Cover 6K working group. It's our pleasure having you here on this very special day. I welcome you, ma'am, on behalf of the organizing committee and Manipal Center for Natural Sciences. Over to you.
Thank you very much for inviting me to give this talk. It is indeed, I'm very honored that you invited me to give this talk, and I'm really very thankful to Dr. Prithviraj, Dr. Vivek, to Dr. Srijit, and all the other organizers at Manipal for putting together this program. So let me start my presentation now. So as Vivek told you, it is, why does the past hold the key to the future? So now this is a very broad topic, and I in fact asked them whether they wanted it this broad, but they seem to like it, and I decided to keep with it. But basically, my expertise, as he told you already, is with pollen grains. And so what I would like to explore is, how does microscopic pollen, this tiny microscopic pollen, how does it help with understanding this past, which is really very important for the future. So in this talk, what I will try to do is actually to introduce you to basic palynology, because I was told that this is a mixed audience and many people may not be very familiar with this particular field. So it will be quite a lot of basics, but I will also try to quickly cover some applications, and also most importantly to talk about how this is actually going to help with our future and with sustainability, topics which were mentioned in the earlier talk today. So I will start with the general background as to why we need to understand. So basically I'm talking in an ecological context and also with reference to our physical environment. So in terms of the past, what has been the evolution? Why it is important to understand this? So we need to have, first of all, a very general background, and then the talk is going to be structured in terms of, with the general background, to go into what is actually pollen biology, and then going from pollen biology to paleoecology, palynology, introducing you to basic morphology, and then how it's applied in past vegetation and climate reconstruction. So basically, this is this is a picture which is showing you the outline of the monsoon. Nowadays, this picture is actually very familiar for you, even from the newspapers, because we are having quite a lot of issues with the monsoon, in the sense that we are sensing a lot of change in it, in terms of the intensity, the frequency, where it's coming, when it's coming, and how it's changing in the way it's coming. In the past 5 years, it's very noticeable in the instrumental climatology itself, and even, as I said, even in the newspapers, because you read about sudden floods and then also droughts in different parts of the place, but especially about sudden floods. So this Indian subcontinent is really very unique in being subject to this monsoon, which itself creates a lot of variation in terms of the physical environment in which plants and people have to thrive. So there's a lot of variation of climate, soils, relief, topography, and all that. So if you zoom in on even just the peninsular part of India, you can see how much variation, what a lot of variation there is in terms of first the monsoon, and then also in terms of the microhabitats which are available for plants and for the vegetation and for people to thrive in. So basically, the high variability of natural factors defines this landscape. And is it only the natural factors? If you just look at the record of prehistoric to early historic sites, and these are the map, this is a map of archaeological sites which have been discovered from 2 of the 4 states in India only, which I sourced from the Historical Atlas of South India, which the French Institute has partly put online, and so you can also access to see it. So this is the area which has also been much settled by people for a very, very long time. So when this happens, what does it mean? So the physical environment by itself has a lot of natural variability, and in addition to that, this landscape heterogeneity, which is natural, is augmented by the human impact, which leads to fragmentation and hence changes in the vegetation, the ecological habitats available, and also the modification of this natural landscape heterogeneity. So this is the reason why, and this is because, I mean, I just took India and I took peninsular India because I wanted to narrow in on something which is more familiar to all of us. But this is of course true of any part of the world, because in many parts of the world, you will see that people have settled, and when people have settled, the natural habitats are actually modified, and then we have to try to understand what is the history of the evolution of the landscape that you're seeing today. So this is actually the importance of the past, because to know what was there in the past is probably very important. And I'm really happy that I'm giving this talk after Dr. Vivek's talk, because in Vivek's talk, he has actually highlighted the loss of biodiversity and also the kind of crisis that we are heading to. So topics which are very important. And he also mentioned about climate change, about how that is very much interlinked with this, and how all of this is really so important in understanding. So he talked about the present, what is happening, and for the future, what we should do. But what I'm going to tell you is how these tiny microscopic pollen can help you to reconstruct the past, and how this can be useful in mitigating, or at least trying to mitigate, this crisis which Vivek had highlighted. So we go to the basics now. So in terms of pollen biology, pollination, and ecological studies. So first of all, pollen grains. It's a term which was coined in 1760 by Linnaeus, and it just means dust. And this is something which most of you are familiar with. It's the dust of the flower, which is commonly yellow in most flowers. But then our recent studies in pollination ecology have showed us that a lot of variation in these color of this pollen grain is also possible. It can go from very dull gray to very bright orange. And sometimes, very rarely, we also see some other colors in the pollen which are produced. But it's generally yellow. And this is the microscopic male reproductive load of flowering plants. And this is actually something which many of you might be familiar with. So this is basically the essence of sorry. Okay. So so ultimately you need to have the seed, and for it to germinate to produce the plant, and it's the mature plant which produces the flower, and the flower produces the pollen grains which are found present within the anthers, and the process of pollination ensures that these pollen grains find a receptive stigma and put out a pollen tube and then fertilize the ovule, and you then get the seed, and then you get the mature plant. And this is the cycle of life. And so pollen grains are really very, very key to this cycle of life. So, and what is this pollination? It is just the transfer of pollen from the anther to the stigma, and it can take place. Sometimes it takes place, there is self-pollination is also possible, or it can take place through external agents. And what are these external agents? These external agents can range from wind pollination, which is basically the simplest mode of pollination. So the pollen gets carried by wind current, and in the process, some of it gets deposited on the stigmas and manages to reach the ovules. However, this is very chancy because it can happen, it need not happen, and by the time it's deposited, the pollen might not be viable. So many things can happen. So the compensation for this is that wind pollinated plants produce enormous number of pollen so that some of them will reach their destination. And despite that, there have been studies which talk about the wasted pollen. Now, in our particular context of the past, this wasted pollen is actually what is providing us the tools to understand the past vegetation history, because it is the wasted pollen that gets deposited in sediments and it gets preserved. And I'll tell you in a while why pollen out of everything else is actually getting preserved. But then apart from wind, you can also have a mediator. So I've just shown you a bee, and I've shown you a picture from the internet, because I found it very attractive. So it's just that the pollen grains can be picked up by bees like this and transferred to the stigma. And so this is insect pollination. But there is also pollination by birds, and humans also pollinate, because there are in parts of Karnataka, which is a major vegetable producing destination, hand pollination is carried out for most of our important vegetables, for tomatoes, for okra, for a number of crops. Hand pollination is actually carried out by people. But of course, hand pollination is limited because it's done by the human hands, and it's a kind of modern innovation which people have done, looking at pollinators and looking at the whole process. So insect pollination, going back to the pollen. So you have sticky and often spiny pollen, which are entomophilous, because you need to have the pollen made in such a way that they will actually stick on to the bees, and that the bees will be able to collect them. So this insect pollination, it can be deliberate. There are several insects which actually collect pollen for their food, But there is also the possibility that insects, when they visit the plant, not for the pollen, but they visit for some other purpose, they visit the flower to gather the nectar, to gather moisture, to gather a lot of other things. They might accidentally also have the pollen stick onto them. So all of this is part of the whole co-evolution of the insects and the plants themselves. So it is the immobility of plants which necessitates vectors. And a lot of primitive plants were pollinated by beetles and flies. And in fact, this was a big puzzle which has still not been solved. And the person who puzzled over it is Darwin, as to why wind pollination actually evolved from animal pollination. Because a lot of pollen gets wasted, as I told you earlier. However, insects which are sensitive to environmental vagaries, failures in might favor morphological features which are favorable to wind pollination. So since then, and because of our observations, what we have found, at least in the tropics, is that many of the plants are adapted to multiple modes of pollination. So what we find in what we call as the pollen spectra, which is what we find from surface soils or from our the samples from our sediment cores, is a mix of wind pollinated and insect pollinated plants here. So entomophilous plants could also be shedding pollen into the air just to ensure that there is reproductive success, because for the plant that is the main point. So all the adaptations that it makes to attract insects or to evolve for wind pollination so that it's able to produce a lot of pollen and disperse them, all of it is to ensure reproductive success. So then comes paleoecology. So now how do you define what is paleoecology? So like, you know from the word paleo that it's the ecology of the past. So it's a reconstruction and study of past ecosystems and between organisms and between their environments. Like, this is the aim of paleoecology. But what we use here is the proxies, what we call as proxies. And the environment also can be measured in terms of proxies. So you could also say that it's, as these people have defined some time back, that it's a branch of ecology that studies the past ecological systems, and it also studies the past of ecological systems. But this temporal dynamic is always demonstrated only by using fossils and other proxies. So in a minute, I'm going to tell you what these proxies are. So the proxies in paleoecology are any indirect indicator of the past environment. So it can be biotic, it can be abiotic. It could also be the interactions within that. So you have this whole list of things, from pollen to charcoal to beetles to plant macrofossils to bones, teeth, tree rings, stable carbon isotopes. But you can also have sediment geochemistry, magnetic properties, elemental ratios. So all of this actually is what makes for a multi-proxy, what we call in paleoecology as a multiple proxy approach. So this multiple proxy approach, because you have to think of this tiny pollen reconstructing the past vegetation. Now, when you go into the field with a botanist to identify a plant, they will be looking at so many parts of the plant. You will have to look at the leaves, you'll have to look at how the leaves are, how is the branch, how is the flower, if there is a flower. But if you're just going to look at it when it's not flowering or not fruiting, then you look at so many features of the plant to identify it. So now with the pollen, then of course, or with just one proxy, say pollen or phytolith or something like that, you're only going to get a part of the picture. So it's basically like a hologram. So the more the number of proxies you use, the clearer is the picture that you get. You will get a picture using each proxy with individual proxies, But if you want a more detailed picture, then you need to add the number of proxies. So that is exactly what we try to do in paleoecology. So why is this? Why do we need to be so careful about this? Because we are using just one single or a number of proxies, and there is so much of interaction. It's such a complex system. So given that as it is ecosystem variability and the biotic response to environmental change and so many other factors are involved in this. So the interpretation of the past using this microscopic pollen really needs to be based on very sound evidence. And I think sound material physical evidence which you can get, and one of the good things as ways to do that is actually with palynology. So this is a field which is dealing. So now we are actually going gradually from understanding what are pollen, their role in pollination, to palynology. So it deals with the identification and analysis of pollen grains, but we are not interested in their DNA because we already spoke about it. The DNA transfer because of the pollination and the seed set, that is very important. But here the stress is on the structure and morphology of the pollen based on its wall. So in palynology, we actually don't care about the DNA. And in fact, if the cell contents are gone, it's actually better for us because we can see the pollen grains much better when we look at them through the microscope. So why do we actually study pollen grains? Because we can identify the source plants from these pollen grains. And they are produced and retrieved in large quantities. So this is something which I already told you. In order to ensure reproductive success, quite often pollen production is in large quantities. And not only that, this is in nature, pollen is meant to be dispersed. And so they also have properties of dispersal. So this will give you a basis for having a proxy which can give you a local regional picture rather than something which is just in that place. Like, I mean, I will give you an example of another proxy which I think it was mentioned earlier, phytoliths. So phytoliths, or if you want to think of vegetal fragments or something like that, it's very difficult to interpret really where it came from. And phytoliths, because they are not dispersed, they are a very good indication of what was present in that location. And that is the reason why phytoliths are used a lot in archaeological context, where it's where people actually store grains and other things, say, you know, in burials and in other ceremonies which people perform. A lot of plants and grains are also offered, and so they are very much in that place. So you can really use phytoliths over there because you know that it's in situ. You're not interested in that context in knowing a local regional picture. But if you really want to know what was happening in the surrounding area where you have taken your core, then pollen grains are really very important. Another thing with the pollen is that you have to retrieve this microscopic pollen from sediment samples. And so there is a lot of lab process, and the pollen grains, or at least the part of the pollen grains that we are interested in, the wall, is very resistant to that. So this is just a quick summary of all the different aspects which go into field, laboratory, microscopy. A lot of things go into this understanding of the pollen record and using of palynology. And in summary, in a brief summary of this, there is no way of directly looking at the past, and we can only infer it through proxy evidences. And it's better to use as many of these proxy evidences as possible. And it's also possible with a good understanding of the habitats. That means the present day ecology. Understanding the modern analogs is also quite an important aspect. And then you can correlate the pollen data with vegetation. So what do we need in order to actually use pollen to reconstruct the past vegetation? It is a good understanding of pollen morphology, reference collection of modern pollen grains. So this is actually of utmost importance. Now going on to describing to you the form and function of pollen, which is actually helping us in our identification of these pollen grains under the microscope. So the shape is actually related, as I said, to its mode of pollination. So this is just an example which is showing you how different it can be. And I already mentioned to you that you can have spines or you can have other things which help the pollen to stick on to the insects. But this is grass pollen, which you're seeing here. And this one is actually, it's very smooth. And usually the family of grasses produces a lot of pollen, and it's wind pollinated, or it can be also self-pollinated. So where do the pollen come from, which are helpful for the identification? So the pollen spectrum, if you're taking surface soils, it is mainly coming from aerial deposition. And in lake sediments, it could be from aerial sources, but it can also contain inwash, and it can also contain other secondary pollen. So whenever you're going to a place to collect a sediment core, then you need to make sure what your catchment is, where you're taking it from, and where is the sediment actually deposited. So these are some caveats which you have to use in terms of this method. Now, going on to the features to be actually observed in the pollen, once you have the pollen slide and you are looking at it under the microscope, the first thing that you look at in a pollen is the size. And you measure it at the poles and the equator as it's shown over here. And the second thing you look at is the apertures and their symmetry. So the apertures are actually a long, simple slit, which is called the colpus or the sulcus. And there's also an endoaperture called the pore, which is more circular. And so you can have various different combinations of the apertures. And you should remember that although these pictures look very two-dimensional, so what you have under a microscopic slide is always three-dimensional. But I'll come to that, and I'll be showing you some brief videos which will actually tell you more about that. So the wall ornamentation, which can go from reticulate to striate, but which can also be smooth, like I showed you the grass pollen earlier. So this is also important factors. So this wall is really important because, as we mentioned earlier, they are marked with characteristic features, and they also withstand chemical treatment with a lot of acids and other things. So the sulcus, the colpus, it's colpate. So if you have a single slit like that, elongated one, it's called colpate. So I'm not going to bother you a lot with terminology, but it's just that these don't occur only separately, they occur also together. And there could be 1, 3, 5, any number of that. And then when you observe the pollen under the microscope, you can observe it like it's a cross section, equatorial section we call this, and this one is of Palacum ellipticum, which is actually a very nice Western Ghats wet evergreen forest tree. And this is the same pollen, you're looking at it from the top. That is why I was telling you that this is not two-dimensional, this is three-dimensional. So it's a spheroid. And when you cut the spheroid, this is what you see. And you see here, there is this colpus, there is this pore, and you have the colporate condition. And this is actually the polar view, because you're looking at the sphere from the top as you're looking at a globe. And there, what are you going to see? You're going to be actually able to see, actually this has 4, but we're not seeing it fully here. But when we move the pollen, we will be able to see it. But you're at least seeing that apertures are there in the 3 corners. So this is some of the techniques that we use in so lux obscuritas, yellow pattern, it's actually it's the light and shade, so because we use a light microscope, so if you have spines as it is in this pollen, this is how it will look like. So when the light falls on the top, there will be brightness, and as you go down in the microscopic focus, you will see how it changes. So you will see this. I hope you're able to see the changes in the way you can look at the pattern which is there, and then you can see all the spines. So that is actually giving you the polar view. And then you can also have the opposite. So when you have depressions instead of spines, then it's the OL pattern, which is the exact opposite, and this is how it will look like. So this is actually again a pollen of the same family, Sapotaceae. I think this is Manilkara, and the one which I had previously shown you, that was from Malvaceae, and that is a Sida pollen. So here you're able to see, you are getting the 4 apertures. So I told you that even in the earlier Sapotaceae, it's actually the 4. So now you're able to see that, and you're also able to see this, what we call as the foveolate pattern in the. So the basic assumption that we use in palynology, especially when we are applying it to quaternary studies, which means the more geologically recent period and also the Holocene period, or even in present day studies, like for example looking at pollen which are coming from surface samples and all that, is that the plant morphology and the pollen morphology remained the same. And so we are able to use the modern pollen morphological knowledge and directly. So this has been tested, I mean, by looking at, that's why the modern pollen reference collection is very important. So these are some illustrations of some of the morphological features. We already saw the pollen grains with spines, and this is actually one of the pollen which appears most frequently in a lot of fossil pollen records as well. And this belongs to the family of Asteraceae. So we call it Compositae echinate because we also have other kinds of Compositae pollen. But you can also have pollen with spines which are looking like this. And you can have this perfect triangular symmetry which is coming from the 3-porate Casuarina. And this is one of the fire-resistant plants which is found almost from the top of the Nilgiris up to the edge of the the tropical dry evergreen forest, which is Dodonia. And this Dodonia viscosa pollen is actually 3-colporate. So these are some, and then I wanted to show you something which will make you really appreciate the diversity of pollen morphology. So this is a picture retrieved from a pollen from honey, actually, from processing honey, and look at the wide variety of the number of pollen types which you find over here. So in terms of the morphology, and of course we know. So I can tell you that I'm seeing some Syzygium pollen here, and I'm also seeing some pollen grains of this tree, which is a very common avenue tree. It's called Peltophorum. And then I showed you earlier, Compositae pollen. So you have this Compositae, but you also have coconut pollen here, and you have really a number of pollen grains here. And it would be really nice if our fossil records also gave us this many pollen, but I can tell you that usually the fossil record, you will only find them hidden somewhere over here when you look at the microscope. So that is how it looks. And how do we actually count the pollen? It's a little bit like ecological transects. So under the microscope, we count in line transects, and we measure what is there in the field of view. So, and usually we use 400 times or 500 times magnification for the counting. But for identification, you need to go to 1000 times. So the pictures which I showed you earlier of the Sapotaceae pollen, those are all taken at 1000 times magnification. Now again, this counting, how does it work? So like I'm running this video and I hope that you are able to see it properly. So you go on like that and you actually count what is in the field of view. So when you come to this field, you count only exactly this pollen, right? Okay, so you move the stage micrometer and you do the count and you count exactly what is within the particular field of view. So now you're seeing that you're getting some different types of pollen. You've got some stained pollen, and you're also seeing that there's a different type of pollen. So when you stop here, so this field in fact has 1, 2, 3, 4, 5. So this is how in fact we do the pollen counting under the microscope. I'm sorry that this one seems to have got repeated. And these are actually fossil pollen from some Holocene cores in the Kaveri Delta. And so this is how the pollen look like. So they will be totally messed up, but you'll have to somehow figure out that there is this criporate, and that's why you know that this is Canthian, and you will have to find out. So all of this means this reference collection is really important, and studying the reference collection is also really very, very important. So to go a little bit faster now, you can have the pollen type usually doesn't correspond to species. So a lot of plant ecologists will be very disappointed, but it's still one of the best proxies. But it's always good to know what is the limitations. So you can have a mixed hierarchy like this. So you can have a pollen type which is species level, and you can also have two families like Moraceae and Urticaceae combined together. And you can also have genus level like Aerva or Strobilanthes. So all of this, so if you want to correlate some of the species types, pollen types with the species, so this is also a very important work for us to do, because we only get our data is just the pollen type, and the pollen type corresponds to which species. This is also very important, and this is again the modern work is really very important for all of this. So it's not always possible to identify. It's just the summary, and in some conditions pollen preservation is not good, but phytoliths are really complementary and they can be very useful. And you can also have that, you can also know very well what I've been repeating many times, so I'll just go over it. So without a firm botanical foundation, you would only have incongruous results. And so this modern study, knowing the vegetation and knowing the present day modern pollen is really very important. So these pollen vegetation relationships, they are really complex thing. So if you are getting a sediment record from a lake or something like that, you can have overrepresented, underrepresented, and poorly preserved pollen. But how do you actually, and this is an example from actually Navya Raghu's PhD thesis, but we used some modeling, but this is a very simple representation of that. So what you see in yellow is what we got from the surface samples from the pollen records. In the same site, the percentage of the vegetation. So you will see that it's really very different. And in some cases, this picture exactly shows you what I was telling here. You can have overrepresentation, underrepresentation, and you can have poor preservation also. So, and very quickly, I would like to present results from one of the sites that we had looked at. It was called Potapuram Cheru. It was in Srisalam, and this was a very recent record. I mean, it was only a record of going back to nearly 300 or 400 years only, and we calculated And this is what we do actually. Once you have all the pollen details, this is how it's plotted in terms of a pollen diagram. And in this case, what has been plotted is the pollen per gram. But you can also plot pollen percentages, the relative pollen percentages of what you get in the entire spectrum. And this is plotted according to the depth and according to the age. And now how do you find the age? You can find the age by radiocarbon dating the sediment sample. But we have recently found that it's really worthwhile to date the actual, to extract pollen from these sediments and also send them for AMS radiocarbon dating. So that is also a very nice technique to use. And the same data, if we actually try to get the, because this was a recent record, we tried with this to use the recent record of the climatology, especially the rainfall. And we found that there was a really nice correlation between the pollen per gram of Hardwickia and the rainfall by doing some kind of regression with against the rainfall gradient. But this is not the only thing. One of the key markers in that record which I showed you, the pollen record, was this tree called Hardwickia bainata. And what we had recommended in terms of forest conservation is that the overall forest cover in fact remained unchanged, but this particular type species was not very well recovered. And we found that, we said that this could be because of the practice of loping of Hardwickia bainata trees, because it could be affecting the flowering cycle, pollen productivity, and hence the tree. So then I'm going to briefly present to you the resource collections. I've talked about the pollen slide collection. So this is what we have at our institute, and this is what we are trying to build up in terms of the phytolith collection. So the pollen reference collection, which was started by P.H. Guinea and consolidated by Dr. Tanikamani, whose name it bears, the Tanikamani slide collection. It's the biggest tropical collection in Asia, and we are trying to build now a new phytolith collection, and we have reached 1,000 preparations for about 400 species. And of course, I didn't tell you a lot about phytoliths, but I also mentioned to you that phytoliths can be very useful in archaeological contexts, especially, and also as a complement to pollen. And plus, I also told you about the fact that you need to have multi-proxy. And most of our pollen collection, they are interlinked with herbarium samples, so that you can go back. So the past, present, and the future, these interlinked collections also, they are very useful to help to highlight. So we use mainly palynology or pollen morphology, and we try to link it, especially with paleoecology, where we collect sediment cores, where we also take archaeological sections, and to interpret the data that we get, we need modern pollen studies. So we also require to combine this with quantitative vegetation studies, and we also need to combine it with complementary proxies, which are like phytoliths. And in order to understand really about in the tropical context, looking at plant pollinator interactions and trying to identify how much of anemophily is there is also becomes quite important. So basically the whole field goes between a concrete and a virtual reality. And of course, it's a very large teamwork, and this is not possible without all of these collaborators. So I would like to give a big first thanks to them. And I would like to leave you with a few quotations for you to think over. So the first one, it's an ancient Tamil poet, a woman poet, who says, "What we know is just a handful of earth. and what we don't know is as huge as this world. So this is the Tamil poet Avayar who says that it's katrathu kaiman alavu, thalladadu ulagalavu. But there's also another poet from a very distant part of the world, William Blake, who says, to see a world in a grain of sand and heaven in a wildflower, hold infinity in the palm of your hand and eternity in an hour. So these two quotes actually, they speak a lot to me in terms of how we can use pollen to reconstruct, in fact, the whole vegetation history, how we go from the really small to the really big. But I'm pretty sure that a lot of people can have a lot of thoughts of this. Thank you for listening to me. I hope I haven't exceeded my time.
Thank you, madam. It was very exciting, and how interesting it would be to understand the past just from the microscopic particle. So if there are any questions, yeah.
I have a couple of questions. One is regarding, I was wondering if you have large flowering plants, it's like a garden of flower plants. Is it not, is wind transmission is not a very efficient thing compared to the insect transmission?
Yes, that is the question which we are asking. See, wind pollination can be very efficient, but wind pollination also means that the plant invests a lot in producing a lot of pollen and wastes a lot of energy doing that. So that is the reason why, in fact, insects and plants have co-evolved over several millennia. And when you have insects ready to pollinate, then it works much better. But it basically depends, because there's such a huge diversity. So it depends on the context, it depends on the environment, it depends on the wind conditions, and it depends. For example, in a tropical forest, I think Vivek will tell you that very well. Tropical forest with the huge canopy, you're not going to have huge amounts of wind possible. So almost most of tropical plants, in especially the wet high canopy forests, they are insect pollinated or they are animal pollinated.
So like that in the course of evolution. Another question is, for a given flower, all pollens are of same shape and same type? This is a very ignorant question.
No question is. Can you please repeat, sir? Because I didn't quite fully catch it.
If you have a flower, you have shown in the microscope, all pollens are of same shape, same type, or different?
No, they are all of different type. I will show you. I will go back to the slide which I had used to show this. Where is that? Oh, oops. Where was my slide with that? I'm sorry. Yes. So if you can see this, can you see the slide now? So you see, they are not the same at all. As I said, you have this triangular pollen. It seems a little bit folded. So this is what we call as syzygium, which is actually the jamun plant. And this is actually belonging to a herb. This one with the spines, it's a compositate. It belongs to a herb. And this is actually a part of what we call as a polyad, which is actually broken, and it's probably coming from acacia, or it's an acacia type, so it can be Adenanthera also, it could be Albizia also. And this is actually from this is actually a tricolporate reticulate, but you see the reticulation is so well defined. So you see the differences. And then you have something here, which is the palm, Borassus palm. So the coconut is smooth and you have the single sulcus here, and you have the boraces with spines and a single sulcus. So no, the pollen morphology is really very different for different plants. And also the size is different. So you see this tiny thing here is also a pollen, and this big thing is also a pollen, and this one is so much bigger. That's the coconut, which is also.
So how is the pollen shape is recognized by the?
Hello.
Same, how is it recognized by the same over?
I think he is asking about the intra-specific variations.
Okay. So the thing is, I think I have some slides here, which probably I will escape and I will go back. I have these. Okay. So basically, you see, we identify pollen grains as pollen types. So Olea glandulifera is a pollen type for us. Justicia is a pollen type for us. Strobilanthes is a pollen type for us. And we have a description of the morphology about how it looks. And we know that a lot of pollen grains from Moraceae and Urticaceae, they share a similar morphology. So these are all pollen morphotypes for us. And then, because we use the reference collection, you know that Justicia type is a morphotype which is shared by Justicia, Rungia, Rostellaria, Acestacea. Similarly, Strobilanthes is a type which is a mix of Strobilanthes, Mackenzie, Nilgirianthus, Dicliptera, a lot of other things. And Croton type, for example, is also all of this. So,
Anupama,
yeah.
Let me intervene.
Yeah.
What he is trying to say is, suppose there is a hibiscus flower.
Yeah.
And pollen is coming from some other jasmine. How that will pollinate? It can never pollinate.
No, it can never pollinate.
How it can recognize? See, that is
yes,
biological, biological, and that kind of a question.
Yes.
It doesn't have anything to do with the shape of the pollen.
Okay, I got it.
Biochemical, biochemical kind of a reaction may be taking place.
Yes.
A kind of a sperm coming from a monkey cannot integrate a dog or a cat or whatever. So
yes,
it's a different species altogether. Something like that. Simple.
Yeah. Yes, it will not. They will not be able to do. And in fact, there has been a recent study because people were saying that in tropical areas this can be a big danger, because when you have a lot of diversity, then how do you ensure that plants can propagate? Because there can be insects dropping off pollen from various different plants onto another. But then there has been a study which shows that this diversity in fact helps in a lot of ways. So it doesn't. So they have studied this also. But it's interesting. They have studied on a stigma pollen from various different other species, and they have found which is the one which is actually successful in the pollination, and which is the one which is producing the seed. So usually it is the same species. And even if the stigma has many other pollen, it doesn't really matter.
Hello.
Hello.
I have a small question. I'm also ignorant in this field. You are studying the pollens that are deposited on the ground for a long time.
Yes.
Okay. Maybe several centuries or even more. So, how does, because once upon a time, whatever was deposited was at a height, and the new pollens have been accumulating over and over above.
Yes.
So, is there any vertical variation? So, are you collecting all these, or do you study the vertical variation of pollens? Why I'm asking this question is, for example, if at some 100 feet, may not be 100 feet, 10 feet below, if there are certain kinds of pollens, and on the top there are different kinds of pollens, then the bottom one would indicate that a different species of plants were there at that time.
Yes.
So
Yes.
do you study the vertical variation of the pollens?
Yes, we do study the vertical variation, and in fact, that is what I had shown you in this pollen diagram over here. So this is with depth. So you have the surface here, and it's going down. It's a 1 meter core, so it's going down 100 centimeters, and we had actually radiocarbon dated it, and we found that it's going from AD 1772 up to 2005. That means it is the present, the time when we went and did the sampling. So we got a number of some radiocarbon ages, and we did some extrapolation also. So indeed, yes, we do study this vertical variation, and it's a vertical variation which gives us these what we call as the pollen zones, which shows us that, for example, this aquatic pollen typha has increased a lot over here. And then you have something called xanthium, which is a human impact indicator. You see there's a lot of it on the top, which is closer to the surface, even though this is still a very remote place. And then you also see that this Hardwickia binata. So this is what I had talked about, how it has reduced in the recent past. Even though our present vegetation survey in the forest showed there were many trees, but they were all loped, and so they were probably not producing enough pollen.
I too have a very preliminary question. You have been studying pollen from the lake cores and all that. Is it
Yes.
possible also to find some pollen grains in the nearshore cores? Maybe it may indicate some heterogeneous influences and how things have been.
Oh yes, near shore also we have looked at, because we have analyzed a pollen record recently, which is off the Kerala coast. So where a study was conducted, and we actually recovered a lot of mangrove pollen in that. So it is possible, but see, basically for us to be able to recover pollen, the site, what you're sampling is very important.
Still water.
So you need to have sedimentation which is taking place regularly, and it should be undisturbed.
What about this? What is that? River mouths like estuaries. Is it possible to get some kind of a signature in the estuarine sediments?
Yes, it's possible. We have
Not high disturbing. Does it not have a very high disturbance of our pollen kind of a function?
Actually it will vary a lot. For example, in the Kaveri Delta, we do have some estuarine cores, and though it's not been fully studied, some of those cores, we did test them, and there is some pollen recovery, but you may not get continuous records in such situations. That is the only thing. So you may have quite a bit of the record which is actually washed off. So you're right that there will be disturbance, but if you're able to get some part of the record, and if you're able to get it radiocarbon dated, then
At
it still gives you a partial history.
Yes, some discrete kind of signals we try to get, and then maybe we can correlate it with much more detailed studies taken from elsewhere. Cool. Okay, thank you very much.
Any questions from the participants who are joining online? Okay, so if there are no more questions, I would like to thank you, ma'am. Thank you so much for introducing us to this exciting field of science, historical science, I mean. And I hope they all have enjoyed this session. Thank you so much, ma'am, for joining us and make your presence over here.
Thank you. Thank you all very much. It was a pleasure to talk to you and to also answer the questions.
Thank you, ma'am. And I would like to thank all the participants, and do join us back tomorrow, same time. And we have 2 more exciting sessions coming up, one by Dr. Prithviraj M on integrated science and technology approach to achieve sustainability in water resources. And the second presentation by Mr. A.R. Shivakumar on science and technology and innovation for a sustainable living around us. See you all tomorrow. Thank you. And for your information, all these talks are streamlined in YouTube live. You can watch it live. Thank you so much. And it's also recorded.